Hello.
Hello.
Hi. How are you?
Okay. What on earth? I'm having trouble finding the window you guys are on. Okay, I got too much on the screen.
Oh, yeah.
You know the,
Great.
you know the feel?
Oh, yeah.
Okay.
Okay.
Let's see. Okay.
Okay.
Okay, so Bradley, I don't know,
Yeah.
You got my message?
Oh, yeah, I got your message. So it's, I was looking at that paper and I was looking at the figure that I think it was figure 15 you mentioned.
15.
Yeah.
Put it up. Do you have a copy there? Put it up on the screen.
Yeah, let me share my screen.
Okay.
Let's see. All right. Let's see. This one here. This is I think this is the figure.
Oh, yeah, that one. Yeah.
Okay.
Yeah, what is that?
What do you mean? What does it mean? Oh.
Well, in terms of how jerky the motion is or smooth. Can that be interpreted that way?
Yeah, so this is a sort of a phase diagram of I guess it's of a single cell that's moving back and forth, or measure of the oscillation. So this is where, you have this pattern here, which is the cell that's going back and forth. The red, the black line is the sine wave. It's a sine wave, and then the red line is the actual movement fitted to a sine wave. So if you go up to this figure C, So if you go up to this figure C, it's just basically it's a comparison of position and velocity. So position is it's maybe kind of like displacement, but it's stated differently, where you move maybe about 120 microns in total, 60 in either direction, and then the velocity changes as you're moving. So you get to the end here, and then it speeds up and slows down, and all this here is like the sort of the variation.
Okay, can I ask you to do something?
Yeah.
Very simple. Can you make a similar plot, but instead of velocity, use acceleration?
Okay.
Okay?
Yeah.
And then compare the numbers you get to the numbers that are in Edgar's paper and the paper headed by Smoku.
All right. Which you're going to put those papers in the chat?
Oh, okay. Let me.
I've never seen the figure like that before.
Yeah.
Here, let me get them on. Okay, Edgar is, let's see, new. Oh no, that's not what I want. Edgar, okay. Wrong window. Again, okay, now what we're looking for is Edgar and Pickett heaps. Let's see.
You want to compare the second derivative of
because that's the accelerations that they got and that we got were very large. And I'd like to compare the accelerations to, okay, let's see, it's Edgar and Pickett heaps, I think. Edgar, here we go. Edgar. Oh, come on, you can have that. Edgar, here it is. Let's try this one.
All right.
Okay. Here. No, that's the. Oh, okay. I don't think, no, that's not it. So what's going on video? Let's see. No, that's ultrastructure. Nope. They have one on which they did have movies. Let's see, maybe I can use this thing. Edgar. Movie. See what it pulls it up. Oops. Come on. Edgar. Wrong. Okay, let me pull up our paper.
Okay.
There should be a reference to it. Let's see. Sudoku. Okay, here's the submitted version of ours now on the reference list. Okay, so this is in my papers, so you should at least have a copy of this.
Okay.
Okay, it's the one by Sabunka, Kinematics of Explosively Jerky Diatom Motility. The book is out now, by the way.
Yeah.
Okay, now let's see if I go down to its reference list.
I'm hoping I can get the book online at the university library. Is
It might
available that way?
You might. I don't know. It depends on whether the university paid for it. It's a Wiley book.
Okay.
Okay. The one. Come on, Edgar. Oh, this is it. Diatom locomotion, computer assisted analysis of the system. Edgar. Oh, it's Edgar by herself, 1979.
Oh.
Okay. I think I should have a copy of that. Let's see. Here. Edgar, 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 1979. Edgar by herself, third. I'll get this one. Yes, there it is. Okay, this one. Okay, I'll put this in the chat.
All right.
Okay, where's chat on this thing?
It's down fourth icon to your right. Yeah, so it's like between the hand and the arrow.
Oops, I've closed it. Open. Come on, open chat. There it is. Can I put a file in chat in this?
No, you can't.
I can't.
No.
Oh. Okay, I'll send it to you by email.
Yeah, that's fine.
Okay. Okay. Bradly. There we go. Let's see. Paper. Okay. It's off to you.
Okay, thank you. Yeah. Yeah, it's an interesting looking graph. I mean, I've been thinking about it actually in the context of this next paper that I've been doing on information processing and psychophysics and, kind of thinking about how that's regulated.
Yeah, it could be that the question is whether or not Vascillary movement is smooth. So I think the simplest way to do that is to take the accelerations in the Edgar paper and the Sabunku paper and compare it to those and see if it falls in the range or it's out of the range. Okay?
Yeah.
And that might be worth a short note someplace to settle the question.
Right.
And we may not need high-speed movies at all.
Okay. Yeah, that's true. because we can just
the other thing was let's see, do you still see my screen?
No, I don't.
You don't? Okay, so let me close chat. Okay, here's start sharing. Okay, share the screen.
Yeah.
Now there's another thing which is perhaps worth doing, but I'm not sure how to do this. If you look at, let's see, I need Brownian motion here. Okay, you see my screen now.
Yeah, I see the screen.
Okay, new window. Okay, now what I'm going to do here is simulation Brownian motion.
All right.
Brownian motion physics. Oh, maybe you should look at this one, because here's one in Python, which means it should be hard to reproduce. The code may be available.
Hello and welcome. I'm James Murphy from encoding.io, where after 9 weekly.
We don't need all that. We don't need his pretty face.
Okay, we go ahead. Time. And to preserve the force of all the randomness of Brownian motion going on here, we find that when we look at squares of differences and add them up, all of these.
Okay, that's no good.
Okay.
I came across. Here, this may be it. Okay, here's a simulation of Brownian motion. Can you see it?
Yeah.
Okay, now you see what they got a bunch of little molecules moving around and a big one that's bumped around, and then they show its path.
Right.
Okay? Now, here's my question. If you took the lines between consecutive positions, you could calculate the mean between two consecutive positions. You could calculate the mean velocity and the mean acceleration.
Yeah.
Right?
Yeah.
Okay. Now, suppose you put a grid over this. I want to make a simple claim, and I'm not sure it's true. I'm not sure how to claim it. If you put a simple grid over this, and took the center of the grid instead of the exact point which they're calculating. In other words, each point where the red dot changes directions would go in the middle of a grid point. Would be moved to the middle of a grid point.
Yeah.
Okay? And in a sense, that represents a spatial resolution of the observation.
Yeah.
Okay? Okay, so here's the claim I'd like to make, and I'm not sure it's true. The finer the grid, the bigger the maximum acceleration.
Okay.
Okay? And I think it would be a matter of getting a hold of a program that does something like this and just putting a grid on there. And the idea then is the grid, as I said, each point on this line would get represented. Let me start it over. Reset.
Right. Reset.
Reset. Okay, each point on the line would be in the middle of the grid point. Okay?
All right.
So every time it changes direction in this Brownian motion, the point would go to the middle because that would, for a given resolution, that's the best you can see.
Right.
Whereas I think they're doing some sort of exact calculation here, at least to the resolution of the floating point variable. Okay.
Right.
Are there any actual images of this?
I haven't looked. There must be some. Sure, I'm sure YouTube's probably full of Brownian motion pictures. Yeah, this is simulated Brownian motion. So, okay. So the question. So, do you understand the question? Then, the finer the grid, the bigger the acceleration.
Right. Right.
True or false?
Well,
Okay.
if you're taking a point for each time it changes direction, so you're measuring on the. Would the grid be this? Every cell in the grid be the same size, or would they be different sizes?
Oh yeah, you could take all of these points and put them on a grid and calculate the distribution of accelerations.
Oh, okay.
Okay, and it would have a tail. My claim is that the finer the grid, the bigger the tail.
All right. Yeah.
And I don't know if that's true or not. It's speculation. I can't find it in the literature. Okay?
Yeah.
Now, I wrote to the author of this one, Andrew Duffy, but he didn't answer. So,
yeah.
but we need a program similar to this that simulates Brownian motion. There should be plenty. That one that was on the YouTube might be publicly available.
Oh, yeah. Yeah, I think these are, pretty common. I mean, they're not that hard to make.
Yeah, so simulations of Brownian motion. You shouldn't have to reprogram the whole thing. Just add a grid to it.
Okay, yeah.
Okay.
Right.
Now, the reason I bring this up is that when I did that work with Subunku, we did 890 frames per second. Okay, so it was a time resolution thing. And we did very high spatial resolution. And when we compared it to Edgar's work was done at 10 frames per second. So we went from 10 to 890.
Okay.
And we wrote the paper, we were surprised because we got bigger accelerations than Edgar got. Okay?
Yeah.
So that's why the speculation here, that it's an intrinsic property of Brownian motion. The closer you look, the bigger it gets.
Yeah.
Which is weird.
Yeah.
Okay, but it's just a speculation right now. Right? So we could test it by a simple simulation. Okay?
Yeah. Yeah, that would
Okay.
be, yeah.
And then it kind of fits in with the the other thing we did in the Subunku paper is we actually fit Brownian motion with drift to the data, and that's exactly what the diatom motion looked like. You know what drift is?
Yeah, it's like a stochastic factor that you would add into the Brownian motion.
Well, it doesn't have to be stochastic. It could be a constant force applied to the particle in addition to Brownian motion.
Okay.
Okay? It doesn't have to be stochastic in itself, but it could be. Okay? Deterministic force would give you. In other words, in this case, for example, if the red dot was pushed to the right, for example, which it's not in this case,
right,
that would be Brownian motion with drift. And you would see it bouncing around, but eventually ending up on the right side.
Right.
Okay. So at any rate, that's the problem. I'll do a reset there.
Yeah.
Okay.
Yeah.
Yeah, I was thinking there's some way of just recording this and throwing a grid over it. Though I don't. Yeah, we could throw grids of different fineness right over these pictures.
Right.
Right. If you could recognize the points and get a fine enough picture, I don't know. Let's see. Can I blow this up at all? Let's see. Yes.
Yeah, you can. Expand the.
Yeah, I can expand the image. So one tricky way to avoid programming would just be to take consecutive pictures, or just take a picture after this is gone, isolate the line, and then each time the line changes direction, put that point at the center of a grid point. Okay?
Yeah.
Yeah, the only problem, if you let it run too long, the lines overlap a lot.
Right.
Okay?
Yeah.
So, okay?
Yeah.
So that would be a sneaky way to do it without bothering the program.
There should be a way to, we could use, there's some sort of program, I'm sure, that could be done, that where you can manipulate, yeah.
I've run across a couple of things when I've been working with my optical coherence tomography, and they have, I think they're using with elastography. They're using a weighted least squares method, and then they have another method they're calling the vector method, and they say it's better because it gives smoother results, and it's more accurate, and it's more resistant to noise. So I don't know. If you want to know more about the vector method, I have to figure out what it is.
Okay.
Yeah.
Okay. Okay.
Like
So Brad,
that.
yeah, this guy stopped after a modest number of moves where there isn't too much overlap. So one could possibly isolate the line visually, get rid of all those little circles, and then find the corners on the line.
Right.
And then move them over to a square grid of a given width of the grid, and then calculate the accelerations.
Right. Right.
Okay, so that would be a sneaky way to do it. Okay.
Yeah. Yeah.
Okay. So what's happening in your audience? You got them both? They're all taking exams now, or?
Well, I think that. Well, I know that we have a couple people working on the Diatom movies, more and more data for some of the video that Thomas generated, and we've been chatting in Slack, which I don't have open, but there's been a robust discussion about how to do this analysis.
Oh.
So they've been working on an algorithm called Pix2Pix, which is like a machine learning algorithm where you take the data, you map it to a mask of some type, and then you use that to basically train the model, and then you can create. You can go through your data that you want to analyze, and it'll pull out and they're really kind of trying to capture the motion data. So the last time they did this, it was like the morphology, just doing static morphology and trying to, extract the cells and their positions. Now they're looking at motion. So they're looking at, movies across many frames, and they're looking at that series of motion, and they're having problems with some of the cells where they're overlapping as they move. So in the comedy, you have that problem, but it's something I think they're overcoming. They're just trying out different techniques of segmentation and trying to define, right.
Yeah. When I saw your phase diagram, and it sunk in, that's when I realized my suggestion that we have to have high-speed movies of Vascillary maybe not unnecessary.
Yeah. I think we can probably approximate it. I don't know. I mean, there's always information and actual sampling, but there's so many techniques now for resampling data, especially in the machine learning community where there've been a number of papers in the last few years where they've tried to simulate oversampling. I think it was Mayuk. It could have been Mayuk from a while back. He presented a paper on this where they did really high speed sampling of some video. Like they oversampled the video, and they used some techniques to avoid aliasing, so that they could get a nice representation of this higher frame rate that didn't exist in the actual movie. They just tried to upsample everything, and so yeah. Now those techniques I think are a bit much for us because I think you need really, you need a lot of computing power to pull it off. So, but I think maybe what these guys are doing right now, Ujwal and Asmit and Tharoon, they're doing some stuff on this. I think that'll help. I think we can get some really interesting data out of that.
Okay, one thing that you might want to look at is that paper by Sabunku.
Okay.
Evaluated 8 different methods for finding the centroid of an isolated diatom.
Okay.
Okay, and then we only used the best one. And it was really technical. I don't know how we did that.
Yeah.
I was there for 2 weeks, and we got all that stuff done in 2 weeks.
Oh, okay. Oh, when you visited them? Yeah.
Yeah, I visited him, then went in one ear and out the other. So
Yeah.
but we pulled it off. But at any rate, so we took advantage of the rigidity of most diatom cells and used that to find the best algorithm for finding the centroid, despite the insides being changing and whatnot. Okay.
Right.
And then we use that for the tracking algorithm.
Yeah.
Okay. Now, I don't know if that'll help with the Vascillary or not, but you might want to skim through those methods and see if any look promising.
Yeah, I'll take a look. That would be good.
But it does have a detailed evaluation of 8 different methods.
Okay, yeah. Sounds
Okay.
pretty complete, yeah.
So, okie doke. Okay. Okay, so look, see if you can just do that. I think the quickest thing to do is the phase diagram with acceleration instead of velocity.
Okay, yeah.
And then let's compare the values you get to the values that are in the 2 papers.
All right.
Okay?
All right, that sounds good.
And then, yeah, whether we go for the speculation on the acceleration getting bigger the finer the grid for Brownian motion, you know, the closest I've been able to find in the literature is that true Brownian motion is what they call differentiable nowhere.
Okay, oh, yes, yes.
Okay, but that doesn't tell you what that's a qualitative statement. I haven't been able to turn that into a quantitative statement. Okay, it's differential nowhere. What is the differentiation for a given grid size?
Right.
Okay.
Yeah, I see what you mean, yeah.
Well, isn't Brownian motion considered to be Brown noise?
Yeah.
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 Yeah, this is a two-dimensional. Yeah, what I showed you was a two-dimensional version of it.
Yeah.
And for the diatoms, we have a one-dimensional version if they're moving in a straight line. Okay.
Yeah.
So at any rate, I think that's why I think it will relate back to the diatoms, because we can start to pin down this notion that a diatom actually is diffusing with drift or with a force.
Right.
But if the force is applied within the cell somehow.
Yeah.
The simplest assumption might be that it's the diatom generates an electric force, and the electric force is biasing the motion of the molecules in the race.
Right.
Which is what we did for the simulation. Okay.
Okay.
So, but I think this can all be formalized much better.
Yeah. Yeah, I think so.
Okay. But at any rate, I think I'd really like to settle this question about whether or not Vascillary is smooth or jerky. Okay.
Right.
I think you've shown it's jerky, but let's see if we can let down.
Yeah, we got to unpack that a bit because it's
Yeah. I mean, because you have to consider, okay, what are the measurement errors you've got? For example, could they be the source of the jerkiness in the phase diagram?
Right.
Okay.
Okay.
Okay. So it's a little bit subtle to make sure it's proven one way or the other.
Yeah. Yeah.
To eliminate questions like that. Okay. That's why I'd like you to look at the Sabunco paper, because we were really, I mean, look, we were able to get down to, I think, something like 5 nanometer resolution in terms of spatial location of the diatom. And we did that by assuming the diatom is rigid and finding the best algorithm for finding its centroid. Okay?
Yeah.
So it's way beyond the resolution of the light microscope.
Yeah.
Even though we were stuck with light microscope images. So. Okay?
Yeah.
In fact, I think they were even almost photon limited because they were high frame rate.
Right.
Okay? So now, most of the images you'll get will probably be 30 frames per second.
Right. Yeah.
Okay, but that's why I think if they fall in between Edgar's results and ours, then we can conclude the motion is jerky, as long as any possible artifacts are eliminated.
Okay, now we're going to go over a few items of interest that I found interesting over the past week, as well as leading out of some of our discussions on Slack. I'd also like to cover a couple papers on various topics to end the meeting. So let me share my screen. First order of business I'm going to talk about is this article by Slackermans, and this is it's a blog post actually from Slackerman's blog, and it's called "Understanding Multiple Neighborhood Cellular Automata," and then NMCA. And so these types of models produce complex and robust emergent structures not commonly seen in simpler models. So this is arises out of a conversation about cellular automata and lifelike simulations that we've been having over the past several months. So this is something that is an extension of a traditional Conway's game of life cellular automata. And so we've talked about Conway's game of life in this group. It's a cellular automata that's implemented on a grid, and each grid takes on a state, and the neighboring states determine the state of the interacting cells, and you end up getting these emergent structures like gliders and other types of things that move around the grid, or they flicker in the grid and they look like something that's living. So, we're trying to simulate something that looks like life in this case. And so in this case, we're doing some things this is something that was developed in 2014 when they were experimenting with neighborhood configuration. So these are some images here of some of these cellular automata, and as you can see, they're not grid-based, they're actually particle-based, and they're able to form these patterns. So some of these look very much like things you'd find in biology. This looks like a packing of rod-shaped bacteria. This looks like a chemical reaction. This might look like a microscopy image. So does this. I mean, just looks like it. I mean, I don't know, how you assess whether or not it's actually living, but it looks like it's living. So that's the idea. So these type of models produce complex emergent patterns, often featuring robust local structures similar to solitons. And solitons are these things from physics that people often use to demonstrate complex systems. The unique properties of these structures offer a vast increase in the diversity of resulting phenomena in comparison to single neighborhood cellular automata. So we were talking about random Brownian motion earlier in the meeting, where we're talking about how to maybe constrain Brownian motion, or to show things that emerge out of the patterns of Brownian motion as particles are moving around in this quasi-random way. What kinds of patterns do they show? So these types of structures offer a vast increase in the diversity of resulting phenomena in comparison to single neighborhood cellular automata. So I know that we do a lot of stuff with Morphozoic in this group, and Morphozoic is similar to this in some ways because it has nested neighborhoods. This is something that has multiple neighborhoods, and so this is a little bit different model. So this is implemented in the shader toy example here. So this is an example of MNCA. It's directly applied to a continuous space model. So this is all continuous. These particles are moving around in continuous space rather than a discrete grid. And you can clearly see that there's this pattern formation going on amongst the particles. And then, so for the sake of demonstration simplicity, this is referred to as a two-state discrete model, but of course they're continuous in a lot of implementations. So this post gives a number of details about this model. We talked about the neighborhoods and update functions. So this is traditional cellular automata like Conway's game of life, defines a single group of local neighbors for each pixel and sums the value at those locations. The resulting neighborhood is used to decide how the update functions will determine that pixel's value for the next time step. But unlike those methods, MNCA uses two or more neighborhoods of distinct composition, with each neighborhood assigned to its own update function. So these are overlapping neighborhoods that give different functions or different criterion to the same particle. These updates are accessed in the order they are executed, with later updates potentially overwriting those that took place before them. So in other words, you have neighborhoods that are sort of not concurrent in time, they're executed maybe over time, differentially. So you have one neighborhood at one point in time, another neighborhood at another point in time. and that gives you differences in the way these particles interact over time. And MNCA models often form structures that exhibit individualized, unit-like local identities compared to solitons. These structures are significantly different to those found in other cellular automata, exhibiting incredible resilience as they interact with their environment in a robust and non-destructive manner. So this is talks about soliton formation. They form solitons. They form emergent structures. They have a reaction to attractive and repulsive forces like most cellular automata. They form collective movements and flocking, and they're able to model compressive and elastic interactions, the formation of crystalline lattices, and metamorphosis of structure layouts causing different behaviors. So you see all these different attributes, not only of lifelike things, but also soft materials. And we talked about soft materials earlier. This, of course, there's this metaphor that we're trying to explore between soft materials on the one hand and living structures on the other hand, or biological structures on the other hand. And so some soft materials are biological, of course, but we're talking in this case about inorganic soft materials. And so there are a lot of analogies there that we can make, a lot of drawing comparisons that allow us to come up with a model of something like an embryo or some developing tissue. Okay, so this kind of just walks through some of this. This is a Conway's game of life simulation here. And you can see what Conway's game of life does. You get these moving things in the array, which look like, some of them are what they call gliders, some of them are pulsing, and so you can see that there. And then this is an example of a neighborhood for Conway's game of life. So this is the focal cell here, so it's like a cell in the middle, and then the cells surrounding it all determine its state. So you can have a rule, for example, that says if all of the neighbors are of state 1, the focal cell is of state 0. And so you can see that in this case. We could have another rule that says if a majority of the cells are state 1, then the focal cell is also state 1. So you can have rules like that in different ways. And so this is in Conway's game of life. There are a couple of rules here. Any cell with 1 or fewer live neighbors dies. Any cell with 3 live neighbors becomes alive. So in other words, if your neighbors are alive, you become alive. If your neighbors die, then you die. And this approximates, something like, an epidemiological model where you see the spread of disease. Any cell with 4 or more live neighbors dies. All other cells survive, retaining the state they had in the previous time step. So again, this resembles something like maybe like the conversion of a stem cell embedded in a tissue. And whatever its neighbors are, it takes on that state, that cell type, and if the neighbors change their identity, then the focal cell will also change its identity. So there are a lot of parallels with life in living systems here. So this is a larger than life neighborhood, and this is of course a little bit bigger. This is different than the kind of neighborhood that you see here in the Conway's game of life. This is a larger than life neighborhood. So the family of patterns in this uses neighborhoods of greater size. So they're bigger than this 9 by 9 or this 3 by 3 grid. And the most well-known example is probably bugs, and uses a radius 5 square neighborhood. So what that means is that you can see that it settles down here, and you start getting these cells that are moving around. So let's go over that again. You get these bubbles. So it looks like things are bubbling around here. Then that activity settles down. Then you start getting these things that move around. They're kind of gliding across, or they're moving across. Sometimes you get bubbling again, like these things that are sort of don't know which state they want to be in. And then you get these coherent structures that move around the area. So this is all generated by this bugs neighborhood, and this is a radius 5, so it means that there are 5 cells out from the focal cell in each direction, and then that fills in the square. So what is this, like a 11 by 11 grid? So it's a pretty big neighborhood. And so all of these cells determine the state of the focal cell in some way. And then you have multiple neighborhood cellular automata. So this is a little bit different even from the example of larger than life, where now it's not necessarily the large size of the neighborhood, but you have these multiple neighborhoods in time. So you can see that for this model, you can choose to average things instead of summing things when referencing the neighborhood. So now you're finding the average behavior of your neighbors instead of the sum of the neighbors. And you have these different neighborhoods, different contexts. So I think I like this. This is nice. This is a basic NMCA example. This is a simplistic discrete MNCA. He's manually constructed using 2 neighborhoods and 6 update functions. So this has a number of update functions. So you have the focal cell here with some area around it, and then the neighbors are way out here where the neighbors can be contiguous to the focal cell. And then this is finally using the same 2 neighborhoods with a different set of update functions results in largely different kind of pattern. So this is a different kind of pattern, all right. This looks like a bunch of worms expanding out. You know, I'm just trying to use some sort of broad metaphor to describe this, but it really does look like, actually it looks like a chemical reaction. And so it's filling in the space, the empty space in the image. So you can see that this area is being filled up with this collective movement of agents, or the state of these different with these different cells, and you're getting this different kind, much different kind of pattern than in the other cases. So this is nice, and you can get this. This is the link here. The next thing I wanted to talk about was I think Jesse was mentioned, Johannes Jagger, who is a European developmental biologist. He's works in Paris and Vienna, so he's interested in complexity theory and development. So he does very much in the same space as we are. And so I wanted to point this out to Jesse. This is his Google Scholar profile here for Johannes. This is a number of different papers here that focus on some of the things we've talked about in recent meetings. So this is the dynamic control positional information in the early Drosophila embryo. This is a paper from 2004. Regulative feedback and pattern formation. So this is about positional information, but linking this to pattern formation and feedback. So bioattractors, dynamical systems theory, and the evolution of regulatory processes. From 2014, this is a more recent paper on the dynamic nature of positional information. So you can see that, you know, he's covered a number of interesting topics, especially of interest to this group. So yes, so that's, we might cover some of these. I think I've covered a few of his papers in previous meetings, and we can cover more of these papers in future meetings. Just let me know what you find interesting from that. And again, that's Google Scholar, and look up Johannes Jagger. The next thing I wanted to talk about is this DevoWorm paper. And this is a long-suffering paper, I know. I promise this is our paper that describes the DevoWorm platform. And I know that we tried to do this once before, where we tried to follow up on Google Summer of Code 2020, and we were going to submit this to the Journal of Open Source Software. And of course, it didn't happen because it wasn't really something they were interested in. And so we wrote this paper up, and this is the state of the paper now. I think it's in pretty decent shape in terms of organization. It just needs to be longer. So this is Mayuk, Ujwal, and myself. And then I would like to also include Maynak on this, because he was the Google Summer of Code person for 2021. And so he updated a lot of the DevoWorm software, and so we want to include him on this. But I also want to, you know, maybe update some of the things from his documentation. So I want to add those things in here. I also want to add some more things from some of the talks that I've given on DevoWorm, especially the platform aspect of it, the educational aspect, and we don't really have a lot on that in here. I'm going to have to go through and start adding on to this. So I'm going to be working on this paper. So we need an abstract, for one thing, and I'll take care of that. But if you have any ideas on what you want to contribute to this, you know, I know that a lot of people have contributed to the data science demos, for example, and then we'll have to have a section on that as well. So I think this is going to be you know, we'll try to get it to the length of maybe like a short preprint, and then we can publish it maybe on Archive, or probably on another preprint server, because I'm not quite sure that Archive would. Well, they might accept it. It depends on the length of the paper. So let's keep working on that. I know that I wanted to make sure I had the current state of the paper, and this is actually in the DevoLearn repository. So if you go to DevoLearn, and that will take you into this paper. It's actually paper.md, so it's in a markdown file, and we're going to work in markdown, I think, to do this editing, and we'll see. You know, you can issue a pull request, or you can make a contribution to it by adding in content and then leaving a note on the pull request, or leaving a note on the commit that says that you made this change. So I encourage, maybe if you want to contribute, let me know, and then maybe contribute through the repository. Okay, now I think we'll move on to papers here. I have the major task board, and I don't want to spend too much time on that. Suffice it to say that I just want to bring this back into people's consciousness. We have a lot of these issues floating around, and some of them are very old. This is the DevoWorm preprint one, which has been in action items for a while, and I apologize that I haven't been more proactive on that, but as you can see, we have a lot of things moving, and we have a lot of things like the NeuroMatch 4 tie-in, which I still haven't worked out, but I think we're going to do something for NeuroMatch 4. And NeuroMatch 4 is right at the beginning of December, so we'll do something maybe concurrently with NeuroMatch 4. So keep an eye out for that. Some of these things, like pattern resumption after self-repair in seashells, is a project that we haven't talked about in many, many months, maybe even years. So if you're interested in that, maybe inquire. We also have other things in here that we haven't revisited. We've talked about in the past, but maybe we need to revisit more. So we need to think about these different projects. I encourage people to go to this. This is a task board here in group meetings. So this is DevoWorm group meetings, and this is the project 1. So this is the group meetings major tasks board for 2020-21. And so you'll see a number of different things that we've finished, but also things that are in progress and things that are on hold. And I would like to really kind of focus on the things that are on hold, because some of those things are things we haven't really been able to do, or we're kind of doing recurrently. So there's some opportunity there to contribute, but also in the action items as well, where we need to get moving on it, and we need to get some momentum behind it. So I think that's enough of that. And if you feel like adding an issue, please try to. I think you have to be a member of DevoWorm. I can give you membership, but then you can add in issues. Okay. So finally, I'd like to get to the papers. And we talked about the quantitative Vascillary earlier. There's some papers here that I found interesting this week, and these are on embryonic muscle movements as strength conditioning. And this is interesting. We've talked about embryonic muscle movements in C. elegans. And what happens in C. elegans is you get in the egg, you get the formation of muscle. And as soon as you get the formation of muscle, you start to see sort of this spontaneous twitching that occurs. And it's electrical activity that's sort of generated by the muscle fibers. The cells become muscle fibers, and the synthesia of the muscle start to become active. Now, the thing about muscle in the embryo is it's not necessarily connected to the connectome. So the connectome usually drives the muscle activity, through motor neurons and other, and the synapses that are involved in that. So there's this activation of muscles that are purposeful, I guess, if you want to put it that way, that they move the organism to different places. So, when the C. elegans is making different movements, the muscle is being driven by these motor neurons. But in the egg, in the embryo, they don't necessarily do that. They just kind of twitch spontaneously. Eventually, they'll become wired to the central nervous system, the connectome, but it's not automatic. Now, in some organisms, and we'll see this in some of these papers, those connections are already sort of made in the embryo, especially in the later embryo. And so let's dig into this. So the first paper is Embryonic Motility, Environmental Influences and Evolutionary Innovation. And this was it's an older paper. It's from Evolution and Development in January 2003. This is Gerd Mueller. It's at the University of Vienna. And so this is a paper on embryonic motility. So this is about movement in the embryo. So this, I think, focuses on what organisms. I think this is just a general review here. So embryos do not passively await hatching from their eggs or amniotic containments, but begin active movement very early on in their development. The first muscle contractions in the chick embryo, for example, so these are chickens, they start on the 3rd day of incubation and subsequently assume a characteristic pattern of increasing and decreasing motor activity. So this is actually a case where they, Victor Hamburger and some other people have explored this problem, and they've looked at this phenomena in the embryo. And so this is something that occurs fairly early on in the formation of the muscle, and then sort of allows for this sort of motor activity, but it's not necessarily linked to the central nervous system or any purposeful movement. It's just increasing and decreasing motor activity. It is long known that embryonic motility represents an important epigenetic component of development. And so in this case, when they say epigenetic, they don't mean like at the molecular level. They mean like something beyond genetics. So this is a component of development that the muscle has to be active to start to keep developing. So the electrical activity is actually bootstrapping the development of the tissue and making it stronger. And if you've ever eaten veal, that veal is where they take cows and they don't let them move, and that results in a softer muscle tissue, and then they take that muscle tissue and use it for the veal. And that's a very common thing actually in cows. They want to make sure that, like when they make meat in a culture dish, they have to ensure that the tissue in the culture dish is stimulated with electricity in order to give it the texture of meat. So this is a very important thing in like agriculture, where they try to get the right consistency of the meat, or they modify the consistency by modifying the activity of the muscle. And so this is something that is what they call epigenetic, meaning that it's not driven by the genetics, it's driven by electrical activity and sort of morphological activity. So in vertebrates, active movement of the embryo was required for the correct development of cartilage, bone, and joints, of muscles, tendons, and ligaments. So this isn't just muscle, this is everything in the body, and of connectivities in the central nervous system. So this is basically, a lot of these movements are driving some of these things to development. And if you know anything about how the central nervous system forms, you know that you have neurons that differentiate, and then you get axons that branch out and try to find each other, and then synapses get established. A lot of that synaptic connection is going to be based on some sort of activity. So active movement is actually driving a lot of that process and sort of strengthening the first synapses that form. So we know that this happens because we see the activity, but also when you have disturbances of embryonic movements, this can lead to severe malformation and functional disorders. This is in the form of pathological motor patterns and neurological deficits that are retained into adulthood. So this is an important thing for the development of the organism. But we don't really know, for example, how this occurs, how this sort of comes to be evolutionarily. So even less is known about the possible evolutionary consequences of this type of environment development interaction. So this is an environment developmental interaction, but what are the evolutionary consequences of this? And moreover, how did this sort of emerge in evolution? Is this something that is, we see these spontaneous muscle movements in C. elegans. Is it something that, even going back to C. elegans, is it important? Well, we don't really know. I mean, I guess we kind of can assume that, but I don't think anyone's done those studies. So motor activity of the embryo is influenced by electrical, mechanical, thermal, and chemical factors. So there's an overview from 1960 that's cited here, and it's maybe a little old, but a lot of these type of studies are fairly old. People don't typically do stuff like this anymore, but I find it interesting. It's not just electrical activity, it's mechanical activity, which we kind of figured because it's muscle and it's moving, but there's also thermal and chemical factors involved, and of course chemical factors are important in the muscle and in the physiology, and then thermal factors can increase or decrease. You can think about like how thermal activity influences organisms if they're raised at a low temperature or a high temperature, and it makes a difference. Decrease of temperature, for instance, not only reduces all metabolic activity, but as a consequence also diminishes motor activity. Okay? Also, the intensity of ambient light conditions were recently shown to affect embryonic motility. So this is another thing, that light conditions also play a role. Chicken eggs respond to a 12-fold increase of illumination by resulting in a 220% average increase of embryo movements. So if you expose the chicken embryo to bright light, it moves more and it moves faster. So this is all endogenous activity. This is not necessarily driven by the nervous system per se, but we see a lot of these environmental effects that affect movement. So this is, cartilage and bone formation, in addition to muscle formation, are affected by these movements. The movements actually provide, and why it's important is it provides sort of a set of forces that the tissues are sort of moving against, and it kind of conditions them. So in the case of like meat, what I was saying before about like how when they make artificial meat in a culture dish, they have to keep stimulating it with electricity. This is because this electrical activity mimics these movements, because when the movements occur, they're stimulated by endogenous electrical activity, and then that electrical activity is actually, the movements are actually moving against some force in the environment. They could be a liquid environment, it could be a physical environment like a surface, hard surface, but there's movement against that surface, and it provides mechanical feedback, and then ultimately modulates the electrical activity. So this is why these movements are important. The same reason why if you walk a lot or run a lot, you can build muscle mass as an adult. It's the same type of process. So that you can see the frequency of embryonic movements in the chick over days of development. You can see that there's this period sort of towards the middle end, maybe 2/3 of the way through development, where it maximizes, and then it decreases towards the end of development. So this is this pattern that they were talking about earlier, where you have a large number of movements and then a smaller number of movements. You get this increase at about day 12 to a maximum at day 12, and then another local maximum at day 16, and then decreases. And so this is a picture of molecular mechanisms driving this. So this is driven, this is driven at the morphological level, but it triggers a lot of these molecular mechanisms. And this is an example from chondrocytes, which is the formation of cartilage. And so this is something that where if you apply compression to cartilage or cells that are going to take on a cartilaginous fate, then you get this sort of, it triggers these molecular pathways. It triggers these growth factors like collagen and GAG and MMPs, and these things will actually stimulate the development of chondrocytes and the development of cartilage. Okay, so now, and then this is an example of the effects of a 12-fold increase on illumination intensity and movement frequencies. So this is an example of where the open bars were where they applied the light, the enhanced light source, and the shaded bars are the controls, which did not see an increase in light. And as you can see, this actually follows this other graph here. This is the normal sort of state of affairs where the embryo is generating these spontaneous movements about 2/3 of the way through development. And again, you see the same pattern here, except that in the case of the enhanced exposure to light, the peak of these the peak of movement occurs earlier, but it's also much more frequent. So you can see that there's a sizable effect of light stimulation. Okay. So I think that's it for this paper. And they talk, well, actually, they talk about the origin of novelty in evolution. So there's a neglected issue in evolutionary theory, and that's the origination of morphological novelty. And so this means that, when you have things that are new in evolution, like new types of tissues or new types of morphological structures, how do they get there? And so some of this work might be able to apply to this idea of the origin of novelty, because you can see that you can see how maybe like new types of tissues could form through this process of spontaneous movements and triggering different biochemical pathways. And at the end of the day, you might say evolve some gene for some new cell type, and that cell type goes through this process of being in the embryo, and then the embryo experiencing these forces and spontaneous movements, and then that results in a new type of tissue that gets reinforced in development, and then you have this new tissue. So, at the end of the day, you can get these new types of innovations through this process, and you have to have the genes in place, but the phenotype is realized through these kind of processes. So the other paper I wanted to talk about here is this is in 2006, so it's actually not a newer paper. It's newer than the paper that we reviewed there. This is early effects of embryonic movement, a shot out of the dark. So this is, so the abstract reads, it has long been appreciated that the study of the embryonic chick and ovo provides a variety of advantages, including the potential to control the embryo's environment and its movement independently of maternal influences. So this has allowed people to identify movement as a pivotal factor in the development of the locomotor apparatus, which is where you want to see that the organism is going to be moving a lot in its life history, its lifespan, and it's trying to sort of get ready for that in the embryo. So this is something that people first identify this type of movement as being part of. So the muscles are twitching spontaneously, and they're getting ready to connect to the motor neurons and then become a movement system for the organism. With an increasing focus on the earliest detectable movements, we have exploited the system by developing novel models and schemes to examine the influence of defined periods of movement during musculoskeletal development. So they use a number of drugs to sort of manipulate these movements early on, and they examine the role of movement in joint, osteochondrial, and muscle development. So osteochondrial is it's bone, it's hard, lamellar bone, and also cartilage. Our initial studies focusing on the joint, so it's a joint like something that you flex, showed that AP-induced hyperactivity had little, if any, effect on the timing or scope of joint cavity elaboration, suggesting that endogenous activity levels provided sufficient stimulus and additional mobilizations without effect. So they tried to mobilize this process, but it didn't really have much of an effect. The endogenous activity was enough for it to form. By contrast, imposition of either rigid or flaccid paralysis prior to cavity formation completely blocked this process and, with time, produced fusion of cartilaginous elements and formation of continuous single cartilaginous rods across locomotion or across locations where joints would ordinarily form. So this is interesting. When they enhance these movements, it doesn't really have much of an effect. But in this model system, when you suppress the movements, when you impose some sort of paralysis on, say, the muscle, but this is, of course, in terms of joint formation, so it's the tissues around the joint. This means that you can completely block the process. So, and once you do that, you can actually create sort of a deformed anatomical structure. So this is an interesting result. And in my other group that I work with, we're very interested in critical periods in development. So mainly we're interested in critical periods with respect to the nervous system, with respect to the brain, with development of behaviors and other things like that. But this is also another nice example of a critical period. But you could imagine that in evolution or in some type of scenario where you have changes in the developmental timing of these things that you can watch in real, you could watch this process where these things get decoupled in time and it actually affects the morphology and how it forms. It's a very interesting evolution of development question. So if you're interested in Evo-Devo, this is a very interesting type of experiment. This observation suggests that static loading derived from spastic rigidity can act to preserve joint cavities. So static loading is where you load a joint, like, load it dynamically, you just kind of load it with forces, and then this is derived from spastic rigidity. So this acts to preserve these cavities. So this is a nice set of experiments, and it kind of gets into this. So they talk about critical periods here. Changes in cartilage and bone growth induced by 3-day periods of flaccid immobilization imposed at distinct developmental phases provide support for a diminution in cartilage elaboration in an early phase, and for a relatively delayed influence of movement and osteogenesis, which is the formation of bone, invoking critical periods during which the developing skeleton becomes receptive to the impact of movement. So this means that this critical period is very important to this process, and the timing of it is very important as well. This skeleton becomes receptive to the impact. So if you develop the movement, if you introduce movements later or earlier in development, maybe it isn't as effective. And this is why we see that graph in the last paper, where there's sort of a peak about 2/3 of the way through development of spontaneous movements, because it's actually having the greatest effect there. Immobilization also exerts differential impact along the proximo-distal axis of the limb, which means that it's doing this in a certain anatomical dimension. Finally, our preliminary results support the possibility that embryonic hyperactivity influences the potential for postnatal muscle growth. So this can also affect postnatal growth as well. And so, again, it's all about timing. It's all about sort of the signals that are generated. If you introduce forces, you can trigger molecular pathways which might be persistent over time if you deliver it if you deliver movements, or when you suppress movements. If you suppress movements at the right time or stimulate movements at the wrong time, they can have deleterious effects on this process, or they can retard this process in a way that will result in changes in the morphology. So this kind of goes through this. They talk more about the role of movement in joint formation. So this isn't muscle. This is actually joint formation, and looking at how movement affects the joint formation of joints and different types of bone. So this is a little bit different than the first paper, but it still has the same. It shows sort of the same results. Okay, so I don't think there are any figures in this paper really worth kind of hitting on, but actually I don't know if I can get back to this part here. I just saw something before I closed it. They were talking about critical periods, and again I wanted to point that out, that there's, some evidence points to the existence of critical periods of skeletal development. And our studies evoke later periods of development during which movement begins to exert a dramatic and significant contribution. Clearly, both the mechanical sequelae of movement, which means the sort of the different components in time, and the structural characteristics of these tissues are changing rapidly during development. It therefore seems unlikely that a purely adaptive capacity underpins this relationship. So that's something I'll leave you with on that. And I think that, this is an interesting area. I don't really know. It's something that isn't really heavily studied, but if you're interested, we can talk about this more. So I think that's it for today. I think we've covered several papers. Heard it before when I presented the papers, and then the papers themselves. So if you're interested in any of these, let me know in the Slack. Actually, I'll post these in the Slack, or I can email you the papers. So thanks for attending the meeting. Talk to you later. Have a good week.